Question number 1. GMP is an allosteric inhibitor of all of the following except PRPP synthetase, PRPP glutamyl amino synthetase, IMP dehydrogenase, adenyl succinate synthesis. Correct answer is option D. Adenyl succinate synthesis. Question number 2. AMP is an allosteric inhibitor of PRPP synthetase, adenyl succinate synthesis, both A and B, and all of these. Correct answer is option C, both A and B. Question number 3. The first reaction unique to purine nucleotide synthesis is catalyzed by PRPP synthetase, PRPP glutamyl amino transferase, phosphoriboside glycinamide synthetase, formyl transferase. Correct answer is option B, PRPP glutamyl amino transferase. Question number 4. Free purine base which can be salvaged are adenine and guanine, adenine and hypoxanthine, guanine and hypoxanthine, adenine, guanine and hypoxanthine. Correct answer is option D, adenine, guanine and hypoxanthine. Question number 5. The enzyme required for salvage of free purine bases is adenine phosphoribosyl transferase, hypoxanthine guanine phosphoribosyl transferase, both A and B, none of these. Correct answer is option C, both A and B. Question number 6. Deoxycytidine kinase can salvage adenosine and deoxyadenosine, adenosine and guanosine, adenine and adenosine. Correct answer is option D, adenine and adenosine. Question number 7. Adenosine kinase can salvage adenosine and deoxyadenosine, adenosine and guanosine, adenine and adenosine. Correct answer is option B, adenosine and deoxyadenosine. Question number 8. Salvage of purine base is regulated by adenosine phosphoribosyl transferase, hypoxanthine guanine phosphoribosyl transferase, availability of PRPP. Correct answer is option C, availability of PRPP. Question number 9. The available PRPP is used preferentially for de novo synthesis of purine nucleotides, de novo synthesis of pyrimidine nucleotides, salvage of purine base, salvage of pyrimidine base. Correct answer is option C, salvage of purine base. Question number 10. The end product of purine catabolism in men is inosine, hypoxanthine, xanthine, uric acid. Correct answer is option D, uric acid is the end product of purine catabolism. Question number 11. The enzyme common to catabolism of all the purine is adenosine deaminase, purine nucleoside phosphorylase, guanase, and none of these. Correct answer is option B, purine nucleoside phosphorylase. Question number 12. Uric acid is the end product of purine as well as protein catabolism in man, fish, bird, all of these. Correct answer is option C, bird. Question number 30. Daily uric acid exertion in adult men is 2 to 6 mg, 20 to 40 mg, 150 to 250 mg, 40 to 600 mg. Correct answer is option D, 40 to 600 mg. Question 40. Dietary purines are catabolized in liver, kidneys, intestinal mucosa, all of the. Correct answer is option C, intestinal mucosa. Dietary purine are catabolized. Question 50. De novo synthesis of pyrimidine nucleotides occurs in correct mitochondria, cytosol, microsome, ribosome. Correct answer is option B, in cytosol de novo synthesis of pyrimidine nucleotides occurs. 60. An enzyme common to de novo synthesis of pyrimidine nucleotide and urea is urease, carbamyl phosphate synthase, aspartate transcarbamylase, arginosuccinase. Correct answer is option B, carbamyl phosphate synthase. 70. The nitrogen atom of pyrimidine nucleus are provided by glutamate, glutamate and aspartate, glutamine, glutamate and aspartate. Correct answer is option D, glutamine and aspartate provide nitrogen atom to pyrimidine. 18. The carbon atom of pyrimidine nucleus are provided by glycine and aspartate, CO2 and aspartate, CO2 and glutamate, CO2 and glutamine. Correct answer is option B. Carbon atom provided by CO2 and aspartate of in pyrimidine. 90. Nitrogen at position 1 of pyrimidine nucleus comes from glutamine, glutamate, glycine, aspartate. Correct answer is option D, aspartate. 20. Nitrogen at position 3 of pyrimidine nucleus come from glutamine, glutamate, glycine, aspartate. Correct answer is option A, glutamine. 21. The carbon atom at position 2 of pyrimidine nucleus is contributed by CO2, glycine, aspartate, glutamine. Correct answer is option A, CO2. Question number 22. Aspartate contributes the following carbon atoms of pyrimidine nucleus C2 and C4, carbon 5 and carbon 6, carbon 2, carbon 4, carbon 6, carbon 4, carbon 5 and carbon 6. Correct answer is option D, carbon 4, carbon 5 and carbon 6. 23. The first pyrimidine nucleotide to be formed in de novo synthesis pathway is UMP, CMP, CTP, TMP. Correct answer is option A. UMP is the first pyrimidine nucleotide formed in de novo synthesis pathway. 24. Conversion of pyridine diphosphate into deoxyuridine diphosphate require all the following except ribonucleotide reductase, thioredoxin, tetrahydrobiopterin and NADPH. Correct answer is option C, tetrahydrobiopterin. 25. Aminoterin decrease the synthesis of TMP, UMP, CMP, all of these. Correct answer is option A, TMP. 26. For synthesis of CTP and UTP, the amino group comes from amide group of asparagine, amide group of glutamine, alpha amino group of glutamine, alpha amino group of glutamate. Correct answer is option B, amide group of glutamine. 27. CTP synthesis from CTP form CDP and inorganic phosphate, CDP and ATP, UTP and glutamine, UTP and glutamate. Correct answer is option C, UTP and glutamine. 28. For the synthesis of TMP from dump, a coenzyme is required which is N10-formyl tetrahydrofolate, N5-methyl tetrahydrofolate, N5-N10 methylene tetrahydrofolate, N5-formino tetrahydrofolate. Correct answer is option C, N5-N10 methylene tetrahydrofolate. 29. All the enzymes required for de novo synthesis of pyrimidine nucleotides are cytosolic except carbamyl phosphate synthesis, aspartate transcarbamylase, dihydroorotate dehydrogenase. Correct answer is option D, dihydroorotate dehydrogenase. 30. During de novo synthesis of pyrimidine nucleotides, the first ring compound to be formed is carbamyl aspartic acid, dihydroorotic acid, orotic acid, orotidine monophosphate. Correct answer is option B, dihydroorotic acid.